Is it okay if I record this?
Absolutely.
Yep. Awesome. Perfect. All right. So can I ask everybody to change your name from KJB to whatever that actually stands for, and Kyle with your last name, and so on and so forth. And then Tatiana and Kyle, can I ask you guys to turn on your videos so I can actually see you? Kind of feels more human that way. Awesome. And Pablo, welcome as well. And then hi, Jose. Hi, Zainab. So Zainab is your first name, I presume? Is that Persian?
No, I'm from Pakistan, so I guess maybe, but it's
it's
Arabic
close enough.
Hebrew
It's
also.
sort of along that trajectory. I was going to guess either India or Persia, and it turns out it's exactly in the middle.
Yeah.
That's great. So Zainab, tell me a little bit about yourself. What do you do?
I'm a graduate student in Rob Krumlov's lab. And so our lab studies like Hox genes, which define like body patterning. And so I'm specifically looking at how certain enhancers within the same cluster are affecting either together or independently the regulation for the Hox cluster.
Beautiful. And which species do you work on?
So I've been working with mice, but my lab also works with fish and ESLs, and we have like lamprey and fly.
So, wow. And also fly. Wow. So vertebrates, mammals, and flies. That's very cool. And what stage of your career are you at?
I'm almost done. I should be graduating soon.
Awesome. So what's next for you? What's down?
I do want to stay in academia, so I'm like starting to wrap up my paper and then start looking for postdocs. I want to get my paper out before I start.
And you'll continue on Hox patterning and stuff like that?
I don't know about Hox patterning. I mean, I enjoy Hox, but I know I definitely don't want to stay in mice. It just takes too long.
I don't know if you've read the Human Genome paper back from '99 or something like that, but Lander, let me share my screen and show you something very cool. So I'm not an author of this paper, but I did make a figure for this paper. So I don't know how authorship was determined, but maybe I should have been. But this figure is mine from the Human Genome paper. And what's really cool about that region is that it shows the Hox cluster in multiple, basically what we found was that the spacing in the Hox cluster was in fact enormously conserved between species. And that these were regions where the repeat elements were dramatically devoid, empty, basically. So you can see here, red is interspersed repeats, and then blue is genes. And that's something that I found at the time to be super, super interesting, that somehow there was a conserved spacing. So now that you understand more about the enhancers in the Hox clusters, I'm curious whether you have some more insights that might be relevant there.
Yeah, so we've looked at, specifically there are 3 enhancers that I'm looking at, and these are like retinoic acid response elements. And so at least 2 of them are considered across species, like I've looked at zebrafish and other mammals, but one of them seems to be like, it's not in like zebrafish, but it is in most mammals. So we're thinking it could be like some kind of duplication, and then there's like some kind of, one is acting like kind of a repressor and the other is an activator, but
fascinating.
Yes. Yeah.
Wow. All right. Next we have Camilla Berensen. Is that a German name?
Yes.
Very cool. But I'm guessing you grew up here?
No, I'm from Argentina, actually, and I'm in Argentina right now.
Wow. Qué bueno. So what do you work on?
I'm currently not working in anything in particular because I'm a grad student at the Stowers program. So we are in this module stage of the program where we are basically going through different modules and learning about what's going on in the institute to then choose our rotation labs.
Very cool. And what are you interested in?
It's kind of intimidating, but I'm interested in developmental biology and metabolism. So I found very interesting the first part of your talk where you were trying to understand the relationship between obesity and the genomic landmarks.
Yeah. So we're now doing all of the single cell profiling that I mentioned towards the end between exercise and diet and sort of how they're intervening. And I think there's an enormous amount of information coming up on the interactions between tissues. We're finding how immune cells are in fact controlling the response of multiple tissues, that stem cells in adipocytes. And that's what we had found in the FTO paper, that in fact, the FTO variant was acting in the 3 days of differentiation, just the first 3 days of differentiation of adipocytes into either brown fat or beige fat. And after that commitment, that genetic locus has zero effect. So it's really early development. So it's kind of cool to understand not just, hey, fat now, but also how fat develops and then specializes and then gets fixated into any of those lineages. Very cool. So Juliana Olivia Hug. So where's your name from?
Hello. I think it's Norwegian-ish.
Okay, sounds good.
Not really sure what side my father comes from.
And what about on your mom's side?
She's El Salvadorian,
Oh,
but you
that's
can't
awesome.
see that in my name.
That's great. All right, so tell me about what you work on.
I'm actually an undergraduate senior at the University of Arizona, but since my school is online, I'm from Kansas. I'm working in the Alvarado Sanchez lab, and I'm working on a project to understand stem cell proliferation, and specifically how genes relating to cells outside of the stem cells signal to the stem cell to proliferate.
Oh, wow. So what you're saying is that it's not just one way. You basically are differentiating, and then as the differentiated cells happen, if I take a stem cell that was near my liver and I put it near my neurons, it's now going to receive a different set of signals. And instead of the sort of within cell programs, it's going to run the from outside the cell programs.
That's what we're trying to figure out. And we're specifically looking at the unfolding protein response and the genes and the proteins related in that, and how that might be affecting the regeneration process of planarian.
I have no idea what unfolded proteins would have to do with regeneration.
There seems to be some type of correlation between the genes expressed downstream that relate to the proliferation of how this animal regenerates.
Can you give me more hints as to why this could be the case?
We see that we went through several different screenings of which genes would most likely relate to stem cell proliferation. And through these several screens, we found a set of candidate genes that relate to proliferation. And then when we irradiate the animal, when we get rid of all the stem cells in the animal, we see an increase in the specific gene GRP78, meaning that it might be outside of the stem cell. And it is part of the unfolding protein response. And when we
stem from the irradiation itself, could it just be an artifact of how you're doing the selection, that in fact the cells are responding to radiation, and that's what you're finding with the unfolded protein response?
Well, what we're trying to see is if there's a difference between the genes that we're leading up to RNA sequencing, and we're trying to see if the genes that are come out of the unfolding protein response is similar to the genes during regeneration.
Wow. Okay. I'm puzzled. I still don't see the link. You could, send me some review papers linking the two, and I would love to read more about that.
Okay. Sorry if I wasn't explaining it the best.
No. I just want to read more. It's an entire new area that I'm totally unaware of, so I'd love to learn more. Can I ask Tatiana and Heining to turn on your videos so that it feels more like we're actually in the same room? And Mining, you're next. Tell me about what you do. So Hu, I guess that's a Chinese name?
Hi, I'm Mining. So I'm the first year graduate student here. I used to work in the industry, so like we develop long-acting insulin, and now I go to graduate school. Okay,
very cool. So tell me about insulin. What did you work on?
We are trying to develop the long-lasting insulin. So so between meals, so we still need a certain level of insulin to keep the blood glucose level low. And so that's what we are developing. And so we are trying to express a conjugation, express a fusion insulin which fused to the Fc part of the IgG, so which will extend its half-life. So, but we need to keep it, the activity is at a optimal level, so it won't be overactivated.
That sounds fantastic. Very cool. Tatiana, Gaitan, you're next.
Hello, sorry, I was eating lunch. That's why I didn't turn on the camera.
This is an actual lunch, so it's completely okay.
Yeah, I'm sorry about that. Yeah, I'm
apologizing for not having pizza in our hands.
right. I'm a grad student in the Seilinger lab, actually. So
where's the name Gaitan coming from?
It is very Colombian. I'm from Colombia, South America.
Good, sounds great.
Yeah, yeah. It's like a side note. It was the last name of one person that was almost president.
Wow.
And it will be the change for our country, but it wasn't. So
well, let's hope that we make a change for our country, right?
Fingers crossed.
Anyway,
Yeah.
Sorry to get distracted. Go ahead. Tell me a little bit about what you work on.
I started in the lab this fall. And I started working in Drosophila, in the early Drosophila embryo, and more specifically in anterior-posterior patterning.
Awesome.
Mm-hmm.
So I'm on Drosophila. Let me actually pull it up. So start in development. So we basically had found that within the Hox cluster of Drosophila, there actually sits 2 microRNAs. So a single Hox locus in Drosophila produces functional microRNAs from opposite DNA strands. And again, this is also work by Alex Stark. What we basically found is that within the Hox cluster of Drosophila, there lies 1 microRNA, miR10, that actually has, the 2 arms both be functional somehow. And then 2 different microRNAs are produced from both strands of the same locus. So one strand produces a mature microRNA and the other strand produces a different mature microRNA, which are controlled by different cis elements. And these are in turn repressing different genes, suggesting this switch function, where when one gets expressed, it represses the others. And when one gets expressed, it represses the others, which is kind of cool. So so I encourage you to take a look at that paper. So Stark and Kelly's and Genes and Development. So
thank you. It looks very interesting.
Very cool. So I don't know if you noticed, but there's a pattern. I found that women speak much less at conferences and at meetings. So that's why I asked all the women first. So, but now all bets are off. So Kylan, you're next.
Kaylin, yes.
I hope we didn't miss anyone. So go ahead, Kylan.
Yeah, just with what I'm working on, is that what you? Yep. So I'm a graduate student, just started my.
Where is that from?
Oh, my name. I think it's Irish.
Okay. Yeah. The AE sounds Irish, all right.
Yeah, pretty far back probably in my family, but they like to say that we're of Irish descent. I don't know how true it is, but that's.
Do you have a little accent?
I'm sorry?
Do you have a small accent from somewhere?
If it's from anywhere, it would be from Indiana, so I hope not.
Oh, that's great. Anyway, so what do you work on?
So I'm starting, I guess, my 3rd year in the graduate school. I'm in Julia Zeitlinger's lab as well.
Oh, that's great.
Yeah, and like Tati, I'm also working in the early Drosophila embryo on a different question. I'm working with a pioneer transcription factor called Zelda, and we're interested basically in the question, how does Zelda overcome an intrinsically high nucleosome barrier for gene activation during the maternal to zygotic transition?
Very cool. Very cool. So what makes it a pioneer, basically?
Well, it binds in the presence of the nucleosome, and it precedes it precedes the binding of basically all of the other important transcription factors at the time, and it's linked very tightly with massive gene activation very early in development. And so basically all of the qualities that people like Kin Zaret have said make a transcription factor a pioneer factor, Zelda fulfills those.
Exactly. No, it wasn't a question, it was a statement basically saying, so what you're actually studying is specifically what makes it a pioneer factor, basically what allows it to bind. Yeah.
Exactly, yes. How does it do it? Exactly.
All right. Jose Fabricio Lopez.
Hi,
Where
I am Mexico.
Mexico, all right. Viva Mexico.
Viva. I am a 5th-year grad student. I was supposed to graduate next year. With the pandemic, I don't know, but I'm happy to love, and I'm working in a phenomenon called meiotic drive.
That's amazing. That's amazing.
Yeah. So I work in Zander's lab with Sarah Sanders.
Yeah.
And she discovered these genes called WTF in yeast.
What a great name.
Yeah. They do really crazy.
WTF before it was fun.
It's still funny after 5 years studying them. And so basically these genes have the ability to spread in the population by being genetic parasites.
Wow.
And I study how they do it, and I do a little bit of population genetics, and then
so what species is that?
Sorry?
What species?
Yeast, fission yeast.
Wow.
Yeah. And the good thing is we can do the experiments. So I get to have asexual generation in a week, and I can do the experiments and see the environmental variables. And so this species tends to inbreed, and that fights back the effect of these selfish elements. And now we're discovering more things that happen at the genome level and association with other genes that kind of impedes the spread of these selfish genes in this species.
That's amazing. So back in 2003, my first paper was actually on yeast. So sequencing and comparison of yeast species to identify genes and regulatory elements. So I used first one. And one of the very cool things that we found was this intine. So there's basically, so the insert, so basically, let's see, the SRVCA gene contains an insertion of 1,400 base pairs that is absent from the 3 related species. The insertion corresponds to the recent horizontal transfer of a known post-translational cell-splicing intine, VMA1. Have you heard about that thing?
Yeah, it rings a bell. Intine, I don't remember what they do, but I remember being in a conference and thinking that they were really cool.
Yeah, the other thing that we found in that paper was this rapid evolution of these telomeres. So basically, if you look at telomeres, there's an enormous amount of gene families that are rapidly expanding. And these are actually environment adaptation genes associated with cell surface adhesion and stuff like that. So that's another, another fun direction to potentially look at.
Yeah, so what is interesting about yeast in general is it's really easy to study any selfish element. So you can study them from what is impacting their evolution through sexual cycles, competition by competing haploid cells.
Yeah.
So yeah, that makes it a really nice organism a model to study evolution in general. And I'm going to check more about that.
The other thing that was kind of fun in that paper was looking at what actually mediates these inversions. And what we found is that with every inversion, you basically had tRNA genes flanking it. And with every translocation, you basically had transposable elements flanking it. So you can think of these mobile elements as not just selfish for themselves, but also as potentially mediating additional evolutionary events.
So what is interesting is that all the WTFs are associated with LTRs, actually.
Yeah.
And we think that one way that they are jumping in the genome is by having those homologous regions and then copying, because when you see the evolution of these genes, their identity percentages is as low as 30%, but they're functional.
Yeah.
Within the species, right?
Yeah.
And they're evolving fastly. And what happens is the rest of the genome, you see the identity is like 99.5%.
Yeah,
so these are in an arms race constantly, and they're evolving constantly. We think that it's just this LTRs are helping them to move around and compete better. So I'm studying how this competition arises when you have two different WTFs. And I am working, like my last project is working how, just with modeling, how these things are evolving.
Very cool. Well, my second paper was also on yeast, and it was looking at this ancient genome duplication in yeast. And in that paper, we basically found that you could actually distinguish ancestral and derived functions between these genes. You could also find replacement and gene conversion. And what we found is that, you know, having that ancestral species that was not whole genome duplicated, you could actually distinguish ancestral and derived functions. So one very cool thing would be to look at sort of, you know, the rapid evolution to see if in fact there appears to be some kind of older function versus rapidly evolving functions that you could distinguish.
Yeah, I have a friend who's studying that, and he has found interesting things. These genes have been recently acquired.
Yeah.
Like 100 million years.
Yeah, very recent. Wow.
Yeah, but they're unique for fission yeast, and that makes it interesting.
Amazing. Awesome. So Kyle, you're next.
Hey, I'm Kyle. I'm a grad student in Cosick C's lab. Yeah. And I am studying potential role of HSP40 or DNAJ family members in the context of long-term memory.
In brain?
Yes, yeah, in fruit flies.
Oh, that's amazing. So we recently had a paper on the formation and the recall of memory. So basically in collaboration with the Thai lab, we basically sorted cells that first acquire a memory, sorted those neurons and profiled their epigenome. And then we sorted neurons that recall a memory, that are activated during the memory recall. And we looked at the epigenomics of those regions. And we found that, in fact, there's a lot of priming and poising with accessibility even before the gene expression changes.
Oh, interesting.
So I can send you that paper if you're interested. It's in Neuron. If you just search my name in Neuron, you'll find it.
Yeah, that does sound interesting.
Yeah.
And what system was that in, by the way?
This was in human.
Okay. Oh, wow.
Yeah.
How do you?
Oh, sorry. I'm sorry. Mouse.
Okay. Gotcha.
Of course. But I meant mammals, basically, not.
The same thing. So.
Yeah. Let's see. Anyway, I can't find it right now. But oh, that's why it wasn't in Neuron. It was in neurobiology. There you go. Neuroscience, Nature Neuroscience. So paper here. Mapping the epigenomic transcriptomic interplay during memory formation and recall in hippocampal engram ensembles. So basically, you know, we carry out these experiments and then sort the cells that respond and then look for their epigenomic and transcriptional changes. So
that's it. Yeah, that sounds really cool. I'll check that out.
Very cool.
By the way, I've enjoyed your conversations on Lex Friedman's podcast.
That's great.
So just out of curiosity, I know you've referenced like human evolution and neoteny a couple times in there. I was wondering if you had any thoughts on like human self-domestication in the context of evolution.
Whoa, you just blew my mind. That sounds super interesting. I've never even thought of the concept.
Oh, interesting. Okay, it's come up in some other stuff I've read, and I just
self-domestication. Are you basically saying that those humans that are weird for society, we kind of kill them off?
Wow. Now I'm gonna have to backtrack and think a little bit. There's an argument that's made that an early part of human evolution in the last X thousand years was self-domestication. So it's known that there's like similarities in domestication across like different kinds of species, like, and neoteny I think is one of those traits that regardless of whether you're domesticating a dog or a chicken or whatever, there's going to be like neoteny and even like weird things like tail curling and that sort of thing.
Yeah, so let me expand a little bit on what you're saying. I think the evolutionary pressure is the opposite. You basically have a natural tendency for things to take longer, but then those animals in the wild that are not able to run within the first minutes of birth are getting eaten up. So with domestication, we're relieving that pressure. We're not selecting for longer neoteny. We're just relieving the selection for short.
Okay. Well, it could be other traits as well, though. Like I think sociability or reduced aggression is part of.
That's sort of what I was referring to. The fact that we kind of weed out the gene pool from people who are just not fitting with the species, basically.
Oh, okay. Yeah. So it would potentially be that kind of thing. Yeah.
Fascinating. But again, what's really phenomenal is if you look at schizophrenia and if you look at, intelligence, I mean, we're basically threading this fine line between intelligence and madness.
Yeah.
That basically all of these genes that were dramatically selected for in human are also associated with all these weird cognitive phenotypes.
Right.
So it's a battle, it's a struggle between the advantage for the species to have these super highly functional humans, even if they don't fit in well, kind of like Newton and stuff, but also to have, an overall cohesive society. And I think that the advantage of sort of the modern drugs, et cetera, is that we allow more of that genetic variation, and perhaps, these super intelligent people who are just totally weird when it comes to social interactions still have a place and still can manage their, psychosis and their weirdness. I mean, I'm thinking of David Nash. I mean, there's plenty of students at MIT who are basically on all kinds of spectra of autism, of schizophrenia, of bipolar, of you name it. And members of my group, myself included, I mean, we're all threading a fine line between, madness and sanity. And, what I love about human societies is, and what I'm talking about at that podcast, is that we want all of that. We don't want 7 billion people who are cognitively normal. We want, the full spectrum of madness and creativity, because that's what causes this horizontal transfer of ideas and the whole society to flourish.
Right. It's the like neurodiversity argument.
Correct.
Yeah.
Correct.
Yeah, that makes a lot of sense.
I'd love to learn more about the stuff that you pointed to. Send me stuff. I now listen to a lot of podcasts. I've never heard a podcast before appearing in one, I have to say.
Oh, okay, nice. Yeah, and I actually have a question related to my project even, but I'll send that to you as well, though.
So every morning I have my headset on, and
yes, same.
When I do anything, I have like 2x speed where I listen.
Yep. Yeah, same.
So yeah, I'm looking for content. So send me stuff. Even if you find just interesting podcasts that I should listen to, I'm happy to learn more.
Okay, definitely. Sounds good.
Awesome.
Thanks,
so AJ, now Trihel, where's your name from?
I've been told it's German.
Yeah, that's what I guess as well.
Okay.
And AJ stands for something?
Yes, Anthony James.
Sounds great.
But my parents wanted to name me AJ, and they decided that would be awkward to write on a birth certificate, so they came up with something for it to stand for.
So it's a backronym.
Yeah, basically, yeah.
All right, take it away.
So I am also a first-year graduate student, so going through the modules and stuff with my classmates all through Zoom. I'm from Minnesota, so the Great White.
Explains the speed of speech. My first student was from Minnesota, and when he went back home, his parents were like, "Matt, you're speaking so much faster now. What's going on?" And every time, I mean, as I was telling Kyle, I listened to podcasts at 2x speed. Every time he spoke, I was like, faster. So anyway, yes, Minnesota.
And so I took a couple years off between undergrad and grad school. And so I worked on neural development, looking at initial myelin sheath formation in zebrafish during my undergrad. And then I worked on gut physiology and gut neuroscience in my post-bac.
I love it. So tell me about gut neuroscience. I mean, what we're doing basically is, as we're studying this whole multi-tissue and multi-organ interactions in obesity and exercise, as I mentioned briefly, what we're finding is all of these neuronal cells in the fat and in the gut. So it's superbly interesting to me to sort of understand that interplay. And, we have tons of cells for you and gene expression changes that are happening in the neurons of the gut as you're exercising. So I'm happy to chat more.
We were focused on actually enteroendocrine cells, if you're familiar with them. So our group was very interested in how they feel mechanical forces and how they transmit that information and what they do to gut physiology.
This is fantastic. Send me references. I'd love to read more.
Sounds good. I can. And my project as a postbac was to use basically optogenetics to turn on enteroendocrine cells and look at their roles in gut physiology.
This is science fiction. This is
I had to figure out how to get a gut to survive outside of an animal. I mean, it was painful, but it was really.
That was during your gap year?
Yeah, that was during 2 years, but yeah.
I mean, I'm sorry, but when people say I took a time off, they go travel in Pakistan or something. I mean, this is not taking time off, my friend. This is like, oh, by the way, before my master's, I did a PhD. This is incredible.
So yeah, I mean, I love the developmental biology. I love neuroscience. I'm very interested in things that are pertinent questions and important. So
when you're looking for a postdoc, let me know.
Sounds good.
Because that diversity and anyway, basically, I mean, for all of you guys, I mean, all your projects, needless to say, sound amazing. So let me know when you guys are ready to graduate and stuff. Cool. So what are you doing now?
So now I'm in grad school, so I just modules and stuff right now.
So I stopped doing research because I'm in grad school.
I am, but right now we're in the genomics module, so I am.
That's awesome.
Learning a little bit of, and probably a small amount, but it's taking me a long time to learn R.
So if you want to hear my lectures, all of my lectures from MIT are online. So any of you guys are welcome to just go watch them on YouTube. I just post them an hour and a half after lecture each time. So, if you want to hear what would MIT teach on this, it's all online.
Sounds great.
Awesome. Carlos Barradas.
Hi.
Where are you from? Or at least where's your name from?
I am from Mexico.
Nice.
I think my last name is probably Spanish.
Okay, sounds great.
Yeah.
Tell me about you.
Yeah, well, I'm in the first year of the graduate school as well. We're dealing with the modules at this moment and learning a lot about genomics.
Yep.
For the last 2 years before the graduate school, I was working on limb regeneration and the axolotl. So the role of microRNAs in regulating the reprogramming events that underlie regeneration.
Yeah.
And I'm very interested in looking at the looking at it from an evolutionary perspective. Every time I think about processes like this, I wonder how it changes in different organisms and why.
Wow, fascinating. So yeah, we've done a lot with evolution. So my field before was comparative genomics and evolution, etc. So we've looked at microRNA evolution in the context of the Drosophila comparative genomics project. And we had, maybe, 9 papers on microRNAs with my postdoc, Alex Stark. But unfortunately, we don't work on microRNAs anymore, but their evolution is superbly interesting because they're just so malleable. Like if you're evolving a transcription factor, you have to figure out how to sort of change the binding site. If you're evolving microRNA, one mutation will do. So,
right, which is also what makes it kind of hard to study or to draw conclusions from. It just seems like they can do anything with a little bit of time to change.
Yeah, convergent evolution is, the norm almost when you have DNA space rather than protein space. That's awesome, super interesting. Ryan Joseph McNamara.
Yeah, so my last name's Irish.
Nice.
And I just graduated my undergrad from the University of Arizona in May. And I actually just started in Dr. Bazzini's lab on Monday as a bioinformaticist.
That's awesome.
So in our lab,
people too, don't.
In our lab, it revolves around codon optimality and its role in influencing mRNA stability. So specifically, the project that I just started working on, we're looking at how that codon optimality sort of mechanism changes between tissues in the body. So in between like liver, brain, cerebellum, we're wondering if one codon like GAC that's normally optimal might be more or less optimal in this tissue compared to another.
I love it. So we have a few papers on the topic. We've basically published a paper with Eva Maria Novoa. Let me mute her. And we're basically looking at sort of codon adaptation index in the context of enormous evolutionary distances as a way to predict the phylum of just a metagenomic sequence just from its codon evolution, sorry, from its codon adaptation index and sort of the codon usage. But I think the whole multi-tissue codon adaptation differences is enormously interesting. But
Yeah.
the way that I would treat it is not think about it as optimal versus suboptimal. You can think of it as another regulatory knob. So if the abundance of tRNAs is actually different globally between liver and brain, which is the example I keep giving for some reason, you could basically look for what is the overall codon adaptation profile for all of the genes in liver and all of the genes in neurons. So build a global chain and then look for how you would predict gene expression to change for every one gene as an additional regulatory knob. So basically, if the RNA abundance for this particular codon is higher, you might even be able to predict that there's in fact a selective adaptation for genes that need to be higher here and lower there, just as an additional control. And I would do that in the context of all of gene regulation. So basically, think about the set of enhancers, the set of promoters, the set of sort of linked elements for every gene as a baseline level of expression, and then variation relative to that, and how much of that can be attributable to this codon level transcriptional rate, translational rate.
Right.
Adaptation.
Yes, someone in the lab actually last year published a paper with a bunch of scores based on a sort of spectrum for the codons. So we know which ones are more optimal than others and everything. And then we're looking at between the tissues, but also between different life stages of the tissues. So the genes that need to be more expressed, say, before birth, might have an influence on which codons are more optimal or less optimal.
Again, I'd love to learn more. Send me some references. I'll read them up. This is super. All right, then we have Haining Jiang and last Pablo.
Yeah,
Is that Chinese, I presume?
Yeah.
Which part of China?
Yeah, I'm still in China, so it's 3 a.m. here. I think it's fun. So I'm as well as a first-year graduate student in Stowers. I just graduated. I just gained my bachelor's degree in biomedical science. So in my undergraduate school, I had 1 year doing the lion-gated ion channels, which is basically doing the electrophysiology day after day. Then after the year, I didn't mean that, but my mentor he transferred, so he transferred to another university, so I had to transfer to another lab. So I'm thinking, so what is interesting? What do I want to do? So then I went to a newly funded laboratory doing epigenetics. Actually, I was focused on chromatin remodelers, which is honestly I can say I know everything about it, but I think it's just interesting, especially in the regulation of transcription. So I'm interested in the utility of computation and genomics in this area. But honestly, I don't have much experience in this.
That's great. So we have a paper that is stalled somewhere because that's, was busy writing 2 different Nature papers in 2 different, completely different topics on mosaicism and on EpiMap. So this is the same student, Carlos Borges, who basically spent maybe a year or 2 looking at chromatin remodelers and general factors and transcription factors in the context of differences between different ENCODE cell types and sort of how they form this modular structure of combinations of factors acting together, partitioning this whole interaction matrix between TFs and their targets and general factors and chromatin remodelers and their targets. So if you know everything about that topic, we would love to maybe get your feedback on that paper at some point if we choose to revive it.
Yeah, of course.
That sounds great. Cool. And then Pablo Guzman Paima, tell me about your name.
I don't know the origin of my name, but I'm Chilean. I'm actually here in Chile. I'm a first year. What? Sorry?
That's crazy. So what my lab manager is Patty Purcell, and it's Patricia, and she's also from Chile.
Oh, cool.
Yeah.
I'm a first year graduate student here at Stowers, so I'm also in the middle of the module courses section of the program.
Very cool.
So I'm currently not working at the lab, but as an undergrad, I worked on neurodevelopment in Drosophila.
Oh, very cool. So what aspect of neurodevelopment?
We were working on, my project was focused on how one signaling pathway, the street-rho pathway, can control two different processes in the lineage development of one cell type.
Beautiful.
We started working on axon guidance, but then we saw that the progenitors of those cells also expressed the same receptor, and I wanted to see if the same receptor was doing two different things at two developmental stages. And
that's awesome. So as I mentioned very, very briefly in my talk, we basically looked at schizophrenia and sort of how, just a sec, I'm trying to think what that paper would be. Oh yeah, let's see. There you go. So we basically have this paper that's currently in review, I think in Nature. Yes, in Nature. But basically what we found is this, so we basically did single cell profiling of schizophrenia and control, and we found this very weird cell type that I mentioned earlier that is differentially expressed. And then we looked at what are the upstream regulators of these genes. And what we found is this module here of, MEF2C, SADB2, and SOX5 and TCF4, which are these four master regulators, which are themselves associated with genetic association with schizophrenia, but also are master regulators of all of the genes that are differentially expressed and that are associated with neurodevelopment as well. So these factors, it turns out, are playing roles both in development and in synaptic plasticity. Once more, in synaptic connection. So once more connecting these two separate processes. So on one hand, there's the neurodevelopmental role, but then if you look at where they peak in expression, there's a second wave where they are also expressed, in later stages. So basically, if you look at early embryo development versus adult, these genes that are sort of found in the middle are kind of active in both for some reason. So again, this whole same concept of sort of taking regulators that are acting in two different places and sort of seeing how are they connected. And I think that's related with a lot of the diseases that we're finding. Basically, they're likely stemming from the interplay between pleiotropic factors that are acting in multiple places and are pulled in multiple directions and somehow perhaps lead to disease through this dual association.
And do you think they're working on similar processes or completely different roles?
Yes is yes. Basically, they have a lot of overlapping targets, but how they're acting individually, we haven't looked so much as to what are the targets of one versus the other versus the other. Cool. We kind of jumped around. If I forgot someone, please wave your hand. If I've spoken with you, give me a thumbs up. Yes, awesome. Good. We are way out of time, but more than general questions, I wanted to sort of get to hear each of you and what you're working on and sort of find points of connection. But even though I'm supposed to be meeting with the faculty now, can you see if there's any questions that you guys would like to ask that are, related to the talk or anything like that?
I have one quick question. When you try to build the references epigenome, do you focus on a specific age of the individual? Because I guess that the epigenetic signature changes a lot during aging.
So, the grant that we're writing now, and very often when we write a grant, it's because we're just doing the research and we want to eventually pay for it, is looking across the lifespan. Every, so basically, in Alzheimer's, we're looking at late life. In schizophrenia, we're looking at sort of middle. In autism, we're looking early. And what we're doing now is a time course across life, across sort of the teenage years, and sort of emergence of adulthood, early adulthood, and sort of, through life, for how is the epigenome and the transcriptome changing at single cell resolution. And we're doing this in the context of both, neurotypic, as well as high PRS, high polygenic risk score, for diseases, to basically see, even before the disease manifests, how are you changing through your life course? And ApoE4 for Alzheimer's provides a beautiful model because it carries half the genetic risk for Alzheimer's. So if you, and it's also common. So if you just go to the neurobiobank at the NIH and you just randomly select individuals, 40% of them are going to have the ApoE allele, the ApoE4 allele. So we're basically now asking for pre-symptomatic changes across individuals, across life, but not in early development. So I think early development would be super interesting as well. We just don't have the right data sets.
I have one question. How hard do you think it's going to be to kind of formalize all this analysis in a way that it gets translated to medicine? So at the end of your talk, you were talking with Julia about that maybe you can kind of converge all these diseases into certain signatures.
Yeah.
And I guess that requires a lot of work from academia, but do you think the private sector is better focused on that kind of a strategy, kind of narrow down the kind of questions that produce?
So being in Boston, we have all of the pharma companies within a stone's throw from MIT. I mean, it's an incredible environment. And basically, Merck is like a block from my house, and then like all of the other major pharma are a block from my work. So that basically means that I have a lot of friends who are in biotech, and some of them are forwarding me emails that are getting passed around in the company, basically saying, "Hey, they're talking about your paper again." So industry is paying attention. So when we publish papers, they get sent around in pharma for new targets, etc. So that's one mode in which I know that they're taking our findings and translating them without us. There's another mode where several companies are basically giving $1 million a year to my lab to work together. And very often we convince them that the thing that they want to do is exactly what we're doing already. And therefore they're just supporting the research with the agreement that we can publish, but they get to have access to the research early on. So that's, that gives me more hope that we can influence and we can impact the, interaction and sort of build sort of ways of channeling all these findings that we have towards therapeutics. I mean, frankly, I want to make an impact in the world, and I don't care if I make a buck out of that. So basically, I'm super excited to sort of see these findings get translated. And we have several industry collaborations where we're trying to do that. And I also participate in an industry program every year where all of these companies come together and we just share data openly, and we talk about targets and we talk about these things. So, it's not something that we're going to do ourselves, but it's something that we can definitely sort of help make sure it happens. But again, the path is long. It's not going to happen overnight. This is, taking years, sometimes decades to develop. Other questions?
I just have like a non-science per se. Did you always wanted to be a PI, and like what do you most enjoy about it? I mean, because you have like so much different stuff going on. How do you keep
strange person. I've never had a plan. I never actually decided to become a professor. I never decided to go to university. At every stage of my life, I was just doing what was fun. And at some point I started doing research, frankly, because my friends were doing research and I loved it. And as I was doing research, I have a very funny story where I gave a talk internally at MIT. And there's a postdoc from Pavel Pevsner's lab. So Ben Raphael was at MIT, and he heard my talk, and he emailed Pavel, who was the department head for computer science, computational biology there. And Pavel emails me saying, we've heard about your work. We'd love to have you interview. So I haven't given a single talk outside MIT. I was just happy doing research, and they're inviting me for a faculty job interview at UCSD. So I did not respond to that email for a year. But exactly 1 year later, I replied without saying, "Oh, sorry for the slow response." I just replied saying, "Hey, Pavel, so happy to hear your email. I would love to interview." So he kind of put the bug in my head that, maybe I should apply for a faculty job. But before, I had like zero plan in my head for a career ever. I was just like, going with the flow. So I'm very unlike most professors. I was just basically having an awesome time, and the opportunity showed up. People were interested, and so when my first Nature paper was accepted, this paper that I flashed was published in Nature 2003, and it was 14 pages, a first author 14-page paper in Nature. It was accepted within 7 days. So we sent the paper. 7 days later, it was accepted with very minor changes. And my advisor basically told me, Manolis, don't think this is normal. This doesn't happen normally this way. So upon acceptance of that paper and remembering that a year earlier, Pavel had emailed me, I basically said, why don't I graduate? So I basically told my advisor, he basically told me, congratulations, Manolis, won't that make a great chapter for your thesis? And I was like, wait a minute. Don't you mean won't that make a great thesis? He's like, oh, yeah, that's what I meant. You just have to write the introduction and conclusion. I'm like, sounds great. So then around the corner, I was like, yes. So I was basically, I had to do a minor, pass my quals, choose a committee, defend my thesis, schedule the defense, write the thesis, and actually defend the thesis. And I had like 45 days to do that so that I could graduate at that cycle. So that basically prompted me from eternal life, grad student, which was my plan. Basically, I just love doing research, to maybe I should graduate. So I graduated and I did a postdoc. That's when I published my second Nature paper on the genome duplication of yeast. And that's when I went back and applied to faculty jobs. And that's how I stayed in academia. So my path is totally non-traditional. So if you ask, well, did you always want to do research? I'm like, no, I just loved science and I loved math and I loved quantitative stuff. And, being at MIT, some of my faculty friends now tell me, I had you as my student in that class. And I as soon as you asked your question, I knew that this guy is different. And so maybe those around me knew. I had no idea. But so basically what I'm trying to tell you guys is, don't worry if you don't have a plan. Don't worry. Like basically many of my friends have a plan and they know what they're going to do. I had no plan. Just do awesome work. Basically, do what you love, work hard, just try to be the best at what you do. And eventually opportunities will come. But when they come, work hard to get them. But basically right now, you're in the midst of a long path. Only worry about that moment in time. Don't overplan it, because you simply don't have all the parameters. There's a lot of uncertainty. I mean, about chaos theory and how sort of things are quite unpredictable if you look far enough into the future. Just do the best you can now, publish well, do awesome research, and then things will come. You'll either find an awesome postdoc or a, awesome industry job or awesome non-traditional science job, etc. So, remember one of my students was like, well, Manolis, you're going to be disappointed, but I'm not going to be applying for faculty jobs. I'm going to go to the NIH. I was like, are you kidding me? This is awesome. Or I'm going to become an editor. I'm like, this is amazing, or et cetera, et cetera, et cetera. So, and many of my students have gone to industry and they're doing amazingly well. I think the whole sort of, academia or bust is ridiculous. I think you should just do whatever is awesome for you. And then, now that you're in grad school, do the best thing you can right now. And eventually you will, Yulia is basically telling me now, hold on one second. So yeah, so that would be my sort of career advice. Basically, make a plan if you're the type of person who makes plans. But if you're not the type of person who makes plans, don't worry about it. It's, you'll make a plan when the time comes, basically.
Thank you.
Awesome. Let me check if Yulia is texting me and sort of saying anything about that. So. Okay, next question, and then I'll check if I have to go or I can answer more. Just one second. Hi, Yulia, we're still chatting with the students, and I'm happy to stop anytime, period. Just text me when it's time to meet with the faculty, and I'll join. Please do send me the link. Yes. Other questions?
Julia, you're muted.
Oh, yeah, you're here.
Yeah, I knew you would not.
This is hilarious. So it's the same link, basically.
No, it's not actually. I just joined the meeting. It's like, well, that's probably the only way to make sure.
Oh, I see, I see. Well, I was texting you. I was well aware that at some point I would have to switch. Okay, guys, I think I have to run.
Sorry.
Thank you.
Thank you.
Everyone, wait. Smile for a group photo. Look at the camera and then smile. Okay, ready? Awesome. Bye, everyone.
All right.
Thank you.
Bye.